All right, hello, biology people. I'm here to begin to introduce just very briefly chapter three. So, I do want to cover the idea of macromolecules and how those macromolecules will be formed. Right? So, just again, a small intro at this point. So, up to this point, you should be familiar with chapter two. You should know all of the idea of the elements, gaining stability via bonding, forming small molecules, um, functional groups like amino groups, like carboxyl groups. You should know sort of all of that chapter two information. So we progress, we get a little bit more complex now. So instead of having small molecules, we start then to combine small pieces to form these large molecules. Just kind of like how you would build a house. You start with small bricks or individual two by fours, and you kind of put them together until it gets bigger and bigger and bigger and bigger. So we get into the idea that we call macromolecules. Macro with an A. Macro with an A implies very large. Micro with an I indicates sort of microscopic, very small. So these are going to be giant molecules, macromolecules, big molecules. They can have atomic mass units, atomic weights, atomic masses of over 100,000, right? So that's a big molecule, right? That's a lot of carbons and nitrogens and hydrogens and oxygens all bonded together. So we get to this big sort of generic macromolecule by putting together a lot of these individual monomers. So kind of like in algebra class, you had variables, monomers like a variable, right? So we put a lot of monomers together, a lot of little units, mono meaning one, a lot of single units put together, eventually get us something very large to the polymer. So polymers are formed by covalent linkages, covalent bonds of smaller units called monomers. Um, we're going to get more specific as we get into the different macromolecules. I'll I won't be so generic with the term monomers, right? I'll give you very specific names for those monomers, but for now, just very generalized here. So, what are these macromolecules? I think you've heard of these, right? And chapter two was all chemistry, and I know this is a biology class, and we're trying to get into biological content, right? But we have to understand these very basic chemistry foundations, right? But macromolecules, kind of now speaking biological terms, we're going to call them proteins. So one category of macromolecule. Another category of macromolecule is carbohydrates, lipids, and nucleic acids. So you have to know all of the chapter two stuff so they can make sense as to how proteins are formed, how carbs are formed, how lipids are formed, how nucleic acids are formed. So that's sort of the justification for learning chemistry, chapter two. So then these molecules, you understand their structure, how, why, where, when, and then their relevance in biology will make a lot more sense. Right? So each one of these will be a specific chapter we get into. That's for later exams. Right now, I'm just going to start the introduction of how the chemical reaction of how each one of these is formed. And it's a similar reaction, yeah, for each. So it doesn't matter if you're a human, a cat, a dog, a fish, a frog, a mushroom, a plant. You're going to be comprised of these four macromolecules, right? So the proteins, nucleic acids, carbs, and lipids. So proteins being the dominant, the most abundant, the most important being the nucleic acids, carbs, and then the smallest piece of the pie being lipids. But these are the categories of different macromolecules. And again, there's a lot of different types of proteins. There's different types of nucleic acids. There's a lot of different types of carbs, a lot of different types of lipids, right? But we'll get to those details in later chapters. So for chapter three, introduction, and for fair game material for exam one, I do want you to know this reaction we call dehydration synthesis. So dehydration synthesis, or the old timers may know it as condensation reactions. I don't hardly hear that. I learned it like that, but I don't hardly hear this phrase used much anymore. This is a much more commonly used phrase, dehydration synthesis. And what dehydration synthesis is, it's the mechanism by which macromolecules are formed. So this is a uh, chemical reaction with the assistance of enzymes, and we'll cover enzymes a little bit later. But enzymes help to create certain types of reactions, right? And dehydration synthesis is like a construction, a formation, a building type reaction. So it's going to take little monomers and connect them together. It's going to form a bond between these monomers. And if we're putting things together, we're building, we're making, we are synthesizing, right? And we're going to make the atomic mass, or I should say the molecular mass, get larger and larger and larger. So again, dehydration synthesis. We're gonna, like when you work hard, you go to the gym, you work hard, you sweat, but hopefully you build muscle, right? That's sort of the idea here. So we're gonna be working, we're gonna lose water, and the process we're gonna build something bigger here. Dehydration synthesis. So I'll show you a picture here. So just generic, like a little train car, right? So we have a monomer, and you should remember this O and the H collectively together we call a hydroxyl, right? So we have a monomer with a hydroxyl on one end. We have a monomer with a hydrogen, sort of on some perimeter there, there's a little hydrogen. Well, certain dehydration synthesis enzymes come in and they start sort of instigating, they start sort of breaking and rearranging, and they start to break this hydroxyl and they break this hydrogen. So the enzyme cleaves that bond, the enzyme cleaves that bond, the OH and the H reconfigure to form water. There's the dehydration part. We're removing these two components to form water. And then we have to restabilize. The electrons have to, we got out of balance, so we have to restabilize this monomer, this little train car with this little train car, and we form that covalent bond there. So that's the process we call dehydration synthesis, the formation of macromolecules. And same reaction, this exact same reaction again. We break off a hydroxyl from one, we break off a hydrogen from the other, produces water. To restabilize, the monomer goes and connects with that second monomer there, right? So I hope you understand the significance here, right? So we have three monomers, but they're all bonded together. So this is acting like one thing, like a long train with a lot of cars. It's still a singular sort of aspect. It's still one train. It's got a lot of cars, but it's one train because they're hooked together. So this is one large polymer made up of individual monomers, formed by the mechanism, by the reaction, by the enzymatic process that we call dehydration synthesis here. Same idea, right? Different little, instead of little rectangles, we have circles, but we have a hydroxyl on one edge, a hydrogen on the other. The hydroxyl gets broken, the hydrogen gets broken, and now this circle will come down here to bond with the first two, right? So the dehydration part and then the synthesis part, the building of something larger. So this is dehydration synthesis. Now, the opposite process we call hydrolysis. So hydrolysis will involve a different set of enzymes, slightly different type of reactions, and an opposite goal. So in hydrolysis, we're trying to break polymers into their individual monomers. So this is the opposite of dehydration synthesis. So here we have the big train, right? Big train bonded, 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 bonded. Well, what comes in here now, the enzyme comes and breaks the bond. So we're going to cleave that bond. We are going to break the water molecule. So we break the polymer and then we break the water molecule. So we have four unstable things. So to restabilize all four, right, the O and the H, the hydroxyl, reconfigure to the monomer. So now stable, now stable. the hydrogen reconfigures with the monomer. So hydrogen is now stable, the monomer is now stable, and we managed to break that apart. So the process we call hydrolysis. Hydrolysis, the splitting of water. So we're splitting water and we're splitting the big polymer here. So we do the same thing. We break that bond. We split water. We put the hydroxyl back in place and we put the hydrogen back in place there. So this accomplishes the opposite goal. We're not building, now we're breaking. So this is the breaking, the catabolic reaction, the digestive reaction. So any protein that you ate that hamburger for lunch maybe, this is the reaction that's happening in the digestive system. That protein is being broken apart, sort of little monomer by monomer by monomer by the process of hydrolysis. If you have a cut or scratch that needs repair, it's getting repaired by the process of dehydration synthesis. So at any given moment, we have both types of reactions going on in the body: the repairing, the fixing, the building reactions, dehydration synthesis, and then the breaking, digestive reactions that we call hydrolysis. So same thing here. So we're going to split water. We're going to split that bond, and hydroxyl gets placed on one, the hydrogen gets placed on the other, and we have now a smaller molecular mass for these two than we had when it was all three connected. So with that, that's as far as I want to get into chapter 3. Just understand that when we talk about proteins, carbohydrates, lipids, nucleic acids, all 4 of these are formed by the process of dehydration synthesis. All 4 of those are broken apart by the process that we call hydrolysis. So it's an important reaction. It's a chemical reaction. That's why I'm putting it with the chemistry and all that mess of chapter 2. So, but with that, that's again, that's where we're going to draw the line for chapter 3 for now and for exam 1. So that's all of chapter 2 stuff and dehydration synthesis, hydrolysis, and this little introduction on macromolecules for exam 1. So with that, let me stop this here. And again, lots of stuff for you to look over. But again, this will be the stopping point for exam 1.